Hello and welcome to recent updates in systemic lupus erythematosus presented by Albert Einstein College of Medicine at Yeshiva University at Rheumatology Live. My name is Michael Ayala, and I'll be moderating today's session and programs on Rheumatology Live are interactive. They allow us to take questions in real time throughout this presentation. Now, I encourage you to send us your questions anytime by typing them in the box located at the lower left-hand side of your screen. And I'd like now to take the time to introduce our presenter, Dr. Daniel Wallace. Dr. Wallace is a clinical professor of medicine at the University of California, Los Angeles. He currently leads over 30 clinical trials for patients with lupus and other rheumatologic conditions. Dr. Wallace has conducted pioneering research in lupus, rheumatoid arthritis, ankylosing spondylitis, psoriatic arthritis, and fibromyalgia. His clinical practice is based at Cedars-Sinai, where he is involved in the care of 2,000 lupus patients, the largest practice of its kind in the United States, and where he co-directs the Cedars-Sinai Rheumatology Fellowship Program. Dr. Wallace, I want to thank you very much for joining us today, and I will turn the program now over to you.
Thank you, Mike. We're going to review some of the recent updates in systemic lupus erythematosus. My disclosures can be viewed on this slide. The objectives of the review today is to identify key advances in the understanding of pathogenesis of lupus, to review the new diagnostic criteria, compare it with the old ones, and identify factors associated with end organ damage to patients with the disease. And finally, to discuss the rationale for targeted therapies, especially those that target B cells, and review the status of some of the more interesting and important clinical trials. Almost 90% of my presentation will deal with insights that are less than 2 years old, and so we're going to be talking about new developments in lupus. The first area we're going to go through is pathogenesis, and these are the topics that we're going to review. As far as the ETO pathogenesis of lupus, it appears that a variety of genes predispose one towards lupus, and they're influenced by hormones and the environment. This results in the formation of autoantibodies and ultimately clinical disease due to deposition of circulating immune complexes and the release of pro-inflammatory mediators. Finally, this leads to scarring, damage, and fibrosis. The preclinical disease lasts several years. This is from a New England Journal of Medicine article several years ago, where individuals who were inducted into the armed forces gave blood. And when these patients ultimately developed systemic lupus, several samples from their serum bank were reviewed to look for the onset of autoantibodies. And it appears that 8 to 9 years before the development of clinical lupus, certain autoantibodies such as Rho were present, followed several years later by anti-DNA. So the authors proposed that we have a benign autoimmune phase where patients feel fine and antibodies are being formed, that under the influence of genetics and the environment produces pathogenic autoimmunity and ultimately clinical illness. The pathogenesis of lupus really is twofold from the standpoint of modeling the immune system. First is the adaptive immune system, which is our B cells and T cells, and the next is the innate immune system, which is sort of considered primordial, from primordial times, pattern recognition receptors, which were originally introduced into our organism to fight bacteria and things that were foreign, and produce interferon through toll receptors and plasmacytoid dendritic cells, which initiate an immune response. Both of these are responsible for the release of inflammatory mediators, immune complexes, adhesion molecules, and ultimately vascular targets, which lead to damage. Now, that is the overview. Let's go into what I just mentioned in a little more detail. How important are genes in lupus? Well, we know that the predisposition towards lupus is hereditary. If a woman with lupus has a child, there is a 10% chance that her daughter would get lupus, a 2% chance that her son would get lupus. If she is one of an identical twin, there is a 25% chance that her twin will develop lupus. If she is a fraternal twin, only 2%. So genes in total probably don't account for much more than about 30% of the disease. And many different gene products contribute to increased susceptibility in lupus. This is from Peggy Crow's article in the New England Journal a couple of years ago, where she listed all of the genes that were associated with lupus. And what's interesting is outside the HLA, the most susceptible, the most promising gene susceptibilities only have odds ratios of 1.2, 1.3, and in aggregate, this doesn't account for more than about 30% of all of lupus. So what about the other 70%? Well, the other 70% is related to the environment and something called epigenetics. We know that ultraviolet light, especially UVA, and UVB to some extent, can produce damage when the skin is exposed to it, and we'll show you some slides on that later. There are over 70 drugs that can induce lupus. Certain agents involved in the manufacture of pharmaceuticals, such as hydrazines and tartrazines, can be lupogenic. L-cannabinine in alfalfa sprouts has been associated with patients or in animals developing lupus, as well as a variety of infections through molecular mimicry, and perhaps hydrazines in tobacco smoke, rare reports of certain immunizations, as well as stress factors. But epigenetics is a relatively newly appreciated discipline, and it's what happens. We have our genome, and when the DNA structure is altered but the code is not, we have something called the epigenome, and it's influenced by diet, smoking, and pollution, for example, which then alters the phenotype of our genes. So epigenetics is defined as a heritable or acquired modification of DNA without any change in the DNA base sequence. This is accomplished through three different mechanisms: hypomethylation, histone deacetylation, and microRNA. And these all occur during cell division. They can all interact with each other. And epigenetic modifications underscore the importance of the plasticity or adaptivity of the environment, specifically because they are reversible and hence amenable to pharmacologic intervention. So talking about gender and hormones, 90% of lupus patients are females, and it may be that estrogen is immune permissive, and androgens are immune suppressive, and estrogens might influence T cells to produce inflammatory cytokines. But the major breakthrough in understanding why women represent the overwhelming majority of lupus patients was published in the last 2 years, and that is demonstrating how genes on the X chromosome may upregulate the immune response. This is work published in 2012 from Bruce Richardson's lab at the University of Michigan. What he was able to show that epigenetics is probably responsible for female predisposition to lupus. Transgenic mice were treated with doxycycline that suppressed T-cell endoplasmic reticulum kinase signaling, and this resulted in hypomethylation or even demethylation. And this demethylation of X-linked methylation-sensitive genes on T cells resulted in demethylation of the second X chromosome. And through cell signaling and suppression by environmental agents, this leads to susceptibility of lupus in women.